Oh, tá. Podemos começar então, tá bom?
Bom dia. É sempre muito bom a gente ver gente sentada no chão para uma palestra na universidade. Eu sou Paulo Nussensweig, atualmente pró-reitor de Pesquisa e Inovação. Queria dar as boas-vindas. Como nós temos um convidado que eu acho que não fala português, eu vou passar para o inglês. So, good morning, everyone. It is an enormous pleasure to welcome you to one more of our USP lecture series, in this case, USP Nobel lecture, because we're fortunate to have a Nobel laureate with us today. This series has been organized by the Office for Research at the university for many years now, and it's a great opportunity to listen from leaders in diverse fields of knowledge, to bring them to boost our academic environment here at the university. So it is really my pleasure to be to organize with this series within our office. Very happy to have Dr. Craig Mello with us today. I'm going to pass the speaker to Professor Rodrigo Galhardo, who will introduce our distinguished speaker today. Thank you.
So good morning and welcome everyone to our institute. And it's my pleasure to introduce our guest today, which is Dr. Craig Mello. Craig has had his BS in biochemistry, BSc in biochemistry in '82, and a PhD in cellular and developmental biology at Harvard at '90. For 4 years after his PhD, he went to Fred Hutchinson Cancer Center to a postdoc. And since '94, he's faculty at the University of Massachusetts Medical School, where he's the chair, Blas University Chair in Molecular Medicine, and is working in the RNA Therapeutics Institute. So, and he has been honored with many awards and prizes, and most notoriously the Nobel Prize in 2006. And it's a real pleasure to have him here. His career is prolific in many interesting and seminal advances. He's a Howard Hughes Medical Institute investigator since 2000. And early in his career, when he moved to his independent career, in '98, he published this paper with his colleague and collaborator, Andrew Fire, in which he showed that injecting double-stranded RNA into C. elegans would lead to gene silencing and paved the way for a new field, which is the field of RNA interference, which is one of almost unparalleled in importance because it's was at the time a novel biological entity, very conserved. It has been used widely by many of us as a research tool to silence gene expression, and is also a promising platform for drug and medicine development. So the importance of his discoveries are many. And Dr. Craig Mello was kind enough to letting us know that he was coming to Brazil and offering to come to USP, which we are really, really honored by this offering. And it's a great pleasure to have you here, Craig. And I want to thank you and welcome you.
Thank you, Rodrigo. Bom dia. It's wonderful to be here. So if I could have the lights dim a little bit, I want to introduce the real Nobel Prize winner right here, C. elegans. This very beautiful little organism is about the size of a comma on the printed page. It's very tiny, and yet it's very similar to us. It has muscles like ours. It has neurons like ours. It has only 300 neurons in its nervous system, but it's capable of behaviors. It's also, it can even learn, and very similar to many, the average undergraduate human, it can remember for one day. So it's actually a smart little creature. Its mouth is about the size of a bacterial cell, so it can eat one bacteria at a time, kind of like M&Ms. But it's a really beautiful organism, and you can see the germline here, the ovary in the adult, and basically this little animal has a lot to teach us about ourselves. And the first part of my talk, I'll be talking about why that is, because I think it's just so amazingly cool. The history of life on this planet is so fascinating. And what my research is involved in is how organisms handle information. They have been living in the information age for billions of years. How do they handle information? It's vital to life. Life is information. That's what drives the living system. Every cell in our body is full of information. Information flows through time in the form of DNA and also RNA, and we still have a lot to learn about how organisms are able to manage and maintain information. So I got interested in this question of organisms handling, how organisms handle information, when I read in the newspaper when I was waiting for the school bus one morning at the age of about 17. I read in the Washington Post that scientists had taken the human gene for insulin and given it to bacteria, and the bacteria could read the human genetic code and make the human insulin protein. And of course, this was really exciting because now you could, use bacteria to make insulin for diabetics. You wouldn't need to harvest it from animals. It would be human insulin. But the thing that fascinated me the most about reading this story was that bacteria could read the human genetic code. And to me, that was when, before that, biology just seemed like kind of boring. You memorize all the different parts of the cell, the Golgi apparatus and the mitochondria. And it didn't seem that interesting to me. But when I learned that the bacteria speak the same genetic language as humans after billions of years of separate evolution, that's when I thought, this is amazing. How can that be? Why is that true? And that got me interested in genetics and how organisms handle information. And in this little slide, I want to just give you some perspective on what I think has happened, in the history of the Earth. And much of this, of course, is speculation. Here, what I'm showing you is a timeline. And you can see, the Earth forms about 4.5 billion years ago. That's a long time because the universe is only about 13.7 billion years since the Big Bang. So 4.5 billion years is a really, really, really long time. The Earth is old. Life arose on Earth. The first evidence for cells in the fossil record is about 3.8 billion years ago. So life arose on Earth pretty quickly. And if you think about how this early part of the evolution of life, we call it the RNA world, what was happening there? Lots of little molecular life forms could somehow develop the ability to self-replicate, make more copies of themselves, passing their information, of course, molecularly to their molecular progeny. But gradually over time, what I imagine happened is that these molecular life forms became essentially melded or merged to form the first cell. So multiple little functions, each of which could replicate, presumably had their own little special synthetic pathways that they contributed to begin to form this original cell. And I think that this this coming together, this melding, this symbiosis is really central to how information flows in the history of life and how it continues to flow. But I think almost certainly this coming together of informational entities to form the first cell, and then ultimately to be somehow amazingly, probably very early in the history of life, arrive at a common language, a genetic code that enabled these RNA life forms to essentially create hard copies of themselves and store them in a shared genome, so that now a living cell could express all of these molecular entities. These little life forms that had been living independently could now share their information. Like they built a hard drive and a computer, and then the software could keep running and it could keep updating automatically somehow. Fascinating, incredible. The history of information has to have been something like that. It's really, of course, we'll never know for sure, but it's really interesting to think about. And then, of course, life diversified. It no doubt continued to diversify throughout all of this time. But if you think about the big domains of life, they arose pretty early in the history of life. In fact, right here, you have the cyanobacteria. These bacteria learned how to harvest sunlight. So photosynthesis began on this planet, and the oxidative form of photosynthesis began 2.5 billion years ago. 2.5 billion years ago. Living organisms figured out how to capture energy from the sun, and as a result, began to release oxygen into the atmosphere. And we know this for sure because it created this a major event in the history of the Earth called the oxygen catastrophe. These little organisms, by releasing oxygen into the atmosphere, messed up the atmosphere. They messed it up. Can you imagine an organism messing up its own atmosphere? And what happened was the oxygen reacted with methane that had been keeping the Earth warm and destroyed the methane, which was very abundant in the Earth's atmosphere prior to photosynthesis. That plunged the Earth into an ice age that lasted for 100 million years. This blue is to depict this ice age in this graphic. Anyway, these independent life forms, they survived this 100 million years of ice and continued to, of course, diversify, but they also continued to undergo these kinds of events. So you have the mitochondrial symbiosis, which is, of course, is quite famous because this was, the precedent for eukaryotic cells, right? So melding of life. You have two life forms that then can sort of meld their genetic information to create a new life form. And this is truly amazing. If you look at, this is the living evidence in every cell that organisms can integrate information acquired horizontally, not from their parents, but from an independently evolving cell. They can take that information in, they can rewire it, they can incorporate it seamlessly into their own genetic program. This is truly amazing. The ability of organisms to handle information. All living things are masters of information assimilation, not just from their parents, but from their environment. And so that happened with the mitochondria, it happened with the chloroplast. So you invent photosynthesis here, but you don't, if you're a eukaryotic cell, you don't need to reinvent photosynthesis. All you have to do is meld, fuse with a chloroplast, incorporate a chloroplast into your cell, and now you've got photosynthesis. This is what plants and algae did. Again, melding of life. And I would argue that the only reason we have the complexity and the diversity of life on this planet that you see today is because information flows horizontally. Evolution can occur in every lineage. And when a breakthrough in evolution occurs, something like photosynthesis, that breakthrough can be transmitted across to other phyla horizontally. If we were constrained only to inherit from parent to offspring, the opportunity for life to evolve would be very, very much more limited. And it's very unlikely that we would see the kind of diversity that we see. So the evidence for horizontal transfer of information and the importance of horizontal transfer is obvious in the history of life. And yet it's strange that really you don't learn much about this when you learn biology. It's really something quite fascinating and fundamental. And I could go on and on about this because the history of life is truly, truly, it's stunning. If you look at this event in the history of life is often referred to as the Cambrian explosion. So this is about 540 million years ago. And this is the time in the history of the Earth when for the first time you start to see large fossils. Not actually the first time, but the first time that they were kind of a lot of them. There were a few other faunas back here that were left bigger fossils, but this is the time when animals and plants started to get big. Shortly after the Cambrian, a couple 100 million years later, first land plants, first land animals. Something really amazing happened in the history of the Earth at this point. But it was pretty recent. This is, not that long ago. If you put this on a calendar year, this happened in November 18th, if this is the beginning of the year, November 18th, and the dinosaur extinction is right here. So all the evolution of mammals and the diversification of mammals happened here, and there's a tiny little dotted line at the end, which is about the distance between us and monkeys. But what I want to actually point out is that animals, this short period when animals diversified to occupy land, we often think of as progressive evolution, like, oh, aren't we better? We're so sophisticated. You often hear the word lower organism, like my little worm. People will call it a lower organism. It's not a lower organism. It's just as sophisticated as you and me. Everything alive today is at the pinnacle of evolution, whether it's a bacterial cell, a fungus, or a human. We're all survivors of this incredible history. And the reason that we have this event in the history of life is now well known. This is what the Earth looked like 640 million years ago. This is one of those glaciation events that is called a snowball Earth. And I took this from a NASA website. But the bottom line is, life on Earth has been constrained by the environment, by the essentially the climate controls what's possible for life. And when the Earth looked like this, of course, there was no land, so there were no land animals. But below that ice, there were eukaryotes, animals, metazoans, bacteria that were every bit as sophisticated as the animals and the life around today. So this is a very important lesson. Mechanisms like RNA interference, of course, translation, transcription, splicing, all of those mechanisms, neurotransmission, of course, some of the details like the antibody immune system hadn't evolved yet, but there were plenty of other really sophisticated mechanisms in the creatures that were living below that ice. And so when we discovered RNA interference, we were actually discovering something that was already ancient. We discovered it because we were trying to put information back into the animal, and the animal was responding to the information that we were giving it, because little did we know, but organisms know how to handle information that's acquired horizontally. They're exposed to it constantly. all of us have been exposed to a virus that originated in China a couple of years ago. It spread through the whole world. You saw it. It happens. Information is hurtling around the planet, and it's going between, you know, different organisms all the time. That information ends up in your germline occasionally. There are SARS and coronavirus genetic remnants in the human germline. It's not common for that to happen, but in the history of life over millions of years, it happens constantly. Microbial genomes get integrated into the human genome. There's probably humans out there right now that we don't know who have a microbial genome integrated into their DNA right now at this minute, maybe one of you, and you don't even know it, right? So how do organisms deal with information that is jumping, it's moving around? Well, it's really dangerous, right? For most of us, getting new information inserted into our genome is going to be a disaster, right? From an individual perspective, you're probably going to die. But if you're a species and you don't know how to do photosynthesis, but boy, wouldn't it be useful if I could just sit there on a rock and get energy? You're going to be, you know, maybe the lucky one who integrates that new information successfully into your toolkit of information. And now you're on the trajectory to become the dominant life form, right? So over the history of time, organisms have been thriving constantly on this acquisition of information. Not the individuals, but the species that are better at incorporating new information are the species that are going to win next time there's an extinction. So that's the perspective that you can see in the history of life. And this is why one of the great things, you know, we should all learn this. Information is dangerous, so of course you shouldn't believe it. Don't believe anything you read. Try to disprove everything you read or learn. You know, don't go out there looking for confirmation of what you already believe. These are the kinds of lessons that you need just for handling information if you're a human using the internet. What organisms do, and one of the cool things about RNA interference, is a great way for the organism to capture information and stick it into their genome, but to keep it silent. You can turn it off. You can everybody knows about CRISPR. CRISPR cuts the information, it destroys it. You know, that's partly how bacteria handle horizontal transfer of information. But if you're a bacteriologist, you know that a lot of bacteria will lose their CRISPR. And when they don't have CRISPR, the same species is in an acquisition mode. It can take in new information without CRISPRing it. You know, and later on it can reacquire CRISPR. So again, horizontal information is vital in microbial life, and they have their way of handling it. Eukaryotes have argonauts, and argonauts are silencers, but not destroyers, because they target mRNA. They can slice and cut mRNA, but they can't cut the DNA. They can guide chromatin remodeling, and they can also guide DNA elimination. I won't go into it, but the ciliates do that. But they only do that in the somatic nuclei. They don't eliminate DNA with their argonauts in the germline of the ciliate, the protozoans. Anyway, it's often a tangent there a little bit. But the concept of an argonaut is it's an information handling system that is quite ancient. And here, when we cloned this gene RDE1, so as Rodrigo mentioned, in 1998, we published a paper saying that RNA could induce silencing. Double-stranded RNA could induce silencing, but we had no idea how. And so immediately, of course, with this mechanism. It was so fascinating. We wanted to figure out how it works. So we did a genetic screen starting in 1998. And in 1999, we cloned this gene called RDE1 for RNAi deficient gene number 1. And this was really very exciting because in 1999, we published that RDE1 is related to many genes, including 8 human genes. So there's 4 human genes here, and there's 4 more human genes over here in the Peewee group. So already we can tell, because worms, C. elegans, has homologs of RDE1 also. So RDE1 has worm homologs related to this green clade of argonauts. This is a family tree, if you will, of argonauts. And RDE1 had homologs that are present in both C. elegans and in the human over here, and in C. elegans and in the human over here. And this was really exciting. But what got me the most excited about this story is when we identified the RDE1 gene back in 1999, that mutation completely abolished the response to double-stranded RNA. You could expose animals to double-stranded RNA and they would not do silencing. But what was even more interesting was that among the mutants that we found, RDE2, 3, 5, 6, 7, 8, those mutants were defective not only in the double-stranded RNA response, but they activated the transposons. So transposons, as all of you know, are very common viral-like little elements that are resident in our genome, and they can hop around. But this mutant did not mobilize the transposons, but it completely abolished the double-stranded RNA response. And so the title of our paper, which almost got rejected because it was too complicated, and reviewers hate complications, right? They hate it. But RNAi, we thought already, was related to another silencing mechanism that was silencing the transposons. So we had discovered some genes were required for responding to double-stranded RNA, and other genes were required for responding to double-stranded RNA and for maintaining transposon silencing. And that was really cool. So, of course, the next 20 years, we were trying to find the mechanism that is involved in transposon surveillance. And that turns out to involve these argonauts over here and also these argonauts over here. So when you knock out these argonauts, you mobilize the transposons. You knock out these argonauts, you can also mobilize some transposons. But also, while we're trying to find these, we knocked out these genes. And that was so cool too, because when we knocked out these 2 argonauts in the worm, we got a phenotype that was identical to the phenotype caused by knocking out a tiny gene called LET7 that encoded the first microRNA, or actually the second one, which was identified. MicroRNAs were identified in C. elegans also. And they are little regulatory RNAs that are expressed as a double-stranded RNA. And then they get processed and they get loaded onto these argonauts. Again, homologs of this argonaut. These get loaded with microRNAs, and humans had these. And humans had a LET7 gene. So this was the discovery that this mechanism that responds to double-stranded RNA actually can engage double-stranded RNAs that are expressed by the organism as genes. And now we know that this mechanism is an antiviral mechanism. It's probably one of the ancient functions of this system is to try to control RNA viruses. And so it was responsive to dsRNA, which is common during viral replication. Many viruses are RNA viruses. But the cell uses the same mechanism by expressing hairpin RNAs that fold into double-stranded RNA and trigger a similar response, in fact, using some of the same machinery, but load these argonauts. And so humans and the worm already had these 2 different mechanisms that involve argonauts. They had the microRNA system and they had the transposon silencing system. And I don't want to go into it too much more in detail because I want to talk a little bit more in this lecture about the therapeutic applications, but I definitely want you to realize that there's a lot more to learn about how organisms are using information. And what this system really is very, very simple. It's Google. It's guided search. And how does guided search work? You start with a short query and you use the short piece of information to find bigger chunks of information. And what you want for a search engine is you want to limit the query size. You don't want it to have to be too long. It just needs to be long enough. So to find Shakespeare's play Macbeth, all I have to do is remember to be or not to be, right? And I can get the whole play on the internet. That's how it works. When we discovered this, that was not possible yet. We didn't have the internet yet. And back in the 1990s, we couldn't do that, but now we can. And of course, everybody understands now how important this is. So they're in this protein, which you can see folded up here, this beautiful, this polymer, if you will, of amino acids that folds up into this amazing shape. Show this to your mom. Seriously, you should show it to your dad, whoever is not a scientist. This is what a protein looks like. This part, all of these like structures that you see here, they're made up from just 20 different amino acids that get, put into a chain, and then they fold up into a machine that can do stuff, that has three-dimensional, features and can open up and move around. So this protein holds on to the search query, which is shown here in red, a little red piece of genetic information. And each one of the little steps in this ladder, it's like that spiral staircase ladder, each one of those steps is made up of a nucleotide, and there's only 4 different letters in the alphabet. Life has a very simple language. 4 letters spell 20 words. That's it. It's beautiful. It's very simple. There's no reason that everybody shouldn't know this, right? It's really simple biology. Biology is actually really simple. But what's so cool about this is that the order of the information that's in this red molecule determines the ability of that molecule to base pair with a target, the target shown here in blue. And if the base pairing starts out good, then it continues. The red strand is held tightly by this enzyme on this end. It presents itself for base pairing. It finds a target, then it wraps around the target. The protein opens up a little bit, allows it to wrap, places the target strand here in a catalytic center and can cleave it. And one molecule of this Google can find and regulate all the mRNAs in a cell, turn them all over in a matter of minutes. So this is a really, really powerful information handling system. And when the realization sort of hit the world that wow, organisms use guided search, maybe we can make drugs. There was an incredible amount of hype. This is what happened in terms of like market size, investment, billions of dollars poured into making drugs based on RNAi in the early 2000s, right about the time that we realized that humans had, the ability to do this, work from Tom Tuschl and colleagues. And of course, all of this money led to huge number of clinical trials that all failed. And luckily they failed right after the Nobel Prize. So, but it was a disaster. Nothing worked. It was just absolutely pitiful and sad. And I actually I started a company right about here too, and I hired a really, really great chemist, Anastasia Kvorova. I'll talk about her work in a moment. But the company failed. A few companies survived, but of course, what was wrong was we were injecting RNA into human body, and there's a lot of enzymes that degrade RNA in the human body. So it was just getting degraded. Within minutes, it was getting degraded. And they tried to encapsulate it, and that didn't work. And anyway, it was a disaster. And of course, it was destined to fail. So a lot of hard work had to go into figuring out how to make these molecules more drug-like. And I had hired Anastasia for the company that I started up. I didn't start up any of the companies that survived this. My company basically failed. It switched over to doing small molecules. And so, you know, the good thing was Nobel Prize came here. The university and the state of Massachusetts gave us $100 million because of the Nobel Prize to start something called the RNA Therapeutics Institute. And even though this was happening and all the investors thought that RNA interference was dead, we knew it wasn't dead. We knew that what we needed was better chemistry. We needed chemistry, better stability. We needed to understand how to make these molecules more drug-like. And so we built this RNA Therapeutics Institute, and we named it that, and we felt kind of like, you know, charlatans, because at the time, back in 2008, when we named our department, there was no such thing. There were no RNA therapeutics at all, not one. But we named ourselves the RNA Therapeutics Institute, and I think that's why they gave us the $100 million. Anyway, but eventually these things started to work, and now there are several approved drugs, and it's actually, these drugs are all working in the liver. The liver was the easy target, and they only work because of a lot of chemistry that went into these molecules, but the chemistry is continuing to improve, and I'll talk a little bit more about that. These are some of the winners. Alnylam survived, this company survived, this company survived, and they're all making liver drugs, and they work very, very well. They're not encapsulated, they're stabilized with chemistry, and they're conjugated to this molecule GalNAc, which enables the uptake in the liver. This is what GalNAc looks like, and it mediates receptor-mediated uptake. But what was truly, truly amazing and exciting about these clinical trials in the liver was that, and this is the biggest one, this is for a drug that was just approved in the United States, and it might be approved here in Brazil. It was approved in Europe last year. It's called Leqvio now, but it was inclisiran at the time. And in this trial, what you're seeing here are LDL levels. These are patients who have LDL elevated, high cholesterol, and they're not doing well on statins. So they tried in the context of statin therapy, they tried injecting an siRNA targeting a gene called PCSK9. And that's actually another really interesting story. I have to just segue back for a moment. One of the things that is really exciting, of course, about having an informational drug is that right at the same time that RNAi was discovered, we finished the Human Genome Project, right? So now we had all the information for the genes that are expressed in a human. And as you know, over the last few years, human genetics is just in a huge revolution, because we're learning not only what genes are required or cause disease, we're also learning what genes lead to better health outcomes. So we're DNA sequencing the people who get sick, but more importantly, maybe we're sequencing the people who don't get sick. And so there are a lot of people who can eat whatever they want, bacon, you know, eggs every day, and they don't get heart disease. Zero heart disease in this population of people. And that's because they have a mutation in a master regulator of LDL levels called PCSK9. And so the medicines company and Alnylam teamed up to make drugs that target PCSK9 so that they could lower the activity of this gene, which is naturally mutated in these people who don't get heart disease. And so they did this clinical trial. They conjugated this drug with GalNAc. It goes beautifully into the liver. And what they did here is they gave a dose, and then at 3 months, they gave another dose. But what was so amazing was that the levels stayed down for 6 months. So one dose of this subcutaneously injected into the fat gave silencing in the liver that lasted for 6 months, and one more dose, another 6 months. And I'll tell you that this is now a feature of siRNA therapies in other tissues as well. Long duration of effect, very surprising for a molecule that you would think would be eliminated fairly quickly. What we think is happening is that the RNA, and this is probably why we also had to have stabilization, siRNAs are internalized via the endocytic pathway. They end up in the lysosome. And so if you look at a siRNA labeled in red and you label the lysosome in green, what you can see is that the siRNA gets concentrated in the lysosome. And of course, if this is a harsh environment, RNAs will not be stable there unless they're completely modified. So every linkage in between every nucleotide is modified. Otherwise, these molecules will rapidly be destroyed. But once that's done, this endocytic route probably also creates a depot where the molecules can gradually be released back into the cell, enabling these longer durations of effect. So I think we are benefiting, now that we have better chemistry, we're actually benefiting from this route of delivery. So yeah, so in 2008, we started the RNA Therapeutics Institute. That's the building we built, and my office is here. And we recruited a bunch of people, mainly, most importantly, the chemists, but we also got physician scientists, which I think is really important. We sort of merged ourselves, mixed ourselves up a little bit, got some physician scientists, got them talking to the chemists and the biologists. And that was I think, really, really important because it led to some collaborations that I'll tell you about in a moment. So here's Anastasia and John Watts. There are two lead chemists. And Anastasia, I told you, I recruited her originally to be in my company, but my company failed during the big downturn. And so Anastasia was on the job market, and I recruited her to come to the RNA Therapeutics Institute. And she literally put therapeutics into our, the word therapeutics was meaningless at first, but she's really made it happen. And I'll tell you a little bit more about her work. And John joined us a little bit later. And between the two of them, they set up an RNA chemistry group. You guys should probably do it too, because RNA chemistry is a vast space. And there's a lot of innovation that is still to be done to explore how to make molecules that can enter into these informational mechanisms like RNA interference. And what's so exciting about an informational drug, right? This is a traditional small molecule, and all drugs have properties that determine where they go in the body and what they target, of course. But for a small molecule drug, those two properties that John and Anastasia termed, the dianophore is the property of the drug that determines where it will go. And the pharmacophore is the property of the drug that tells you what the target is, what it will regulate. These two features are really inseparable for most small molecule drugs. So once you have the structure, it determines both of those. But for an informational drug, of course, the dianophore is determined by the conjugate and the chemistry, but the pharmacophore, what it targets, is determined by the nucleotide sequence of the drug. In other words, the informational drug is programmable. Once you achieve delivery to the liver, you can make drugs on any gene that's expressed in the liver. You're off and running. And so the idea that Anastasia and John had is, let's try to broaden the scope of delivery for these kinds of molecules. And this was, of course, where the synergy came in with the physician scientists. So we had physicians in the RTI, the RNA Therapeutics Institute, who had patients, for example, dying from ALS or Huntington disease, brain disorders,
but we had no delivery whatsoever to the brain. And in fact, Alnylam had a program in Huntington, but they closed it down. They couldn't get delivery. But Anastasia was, right down the hall from Neil Aronin, and Neil Aronin's patients were dying of Huntington, and he was tired of it. So he kept bothering her, come on, let's try this, let's try that, let's get it going into the brain. And literally that's, that constant interaction led to a breakthrough that led to this company, which we launched in 2017, a spin out from UMass, that is making an siRNA molecule that delivers absolutely beautifully throughout the brain. It goes into essentially every neuron, every astrocyte and glia. The delivery is stunning. And based on that, we were able to raise $110 million for that company. The company's not publicly traded yet because it hasn't had to raise money. It's got partnerships with Biogen and Roche. It's making, has like 20 clinical programs for Alzheimer's and Parkinson's disease, and Huntington was taken over by Biogen. It's truly exciting. And the story is really amazing behind this. It's a bedside to bench, breakthrough, because again, Neil was like, we got to figure this out. We got to get delivery. And of course they were trying everything they could to try to, attach different molecules to the siRNAs that would make them stay in the brain. They were injecting these molecules into the cerebral spinal fluid, trying to get them to distribute throughout the brain. But whenever they put an siRNA into the CSF, it would be excreted from the CSF within hours. So nothing stayed in the brain. And one of the things they tried was conjugating vitamin D to the siRNA. And when they were making this conjugate, they had a contaminant. They couldn't get rid of the contaminant. So, the mice were waiting. So they went ahead and injected this contaminated mix. It had vitamin D conjugated to an siRNA. This is an siRNA right here. And what happened was the linker, which was supposed to attach vitamin D, attached to another siRNA. So they had these little dimeric siRNAs. That was their contaminant. They injected this mix into the brain, and they got amazing delivery. And of course, they thought it was the vitamin D, but then they purified it, and it was this. So again, the space, chemistry space is vast because it's somewhat empirical. You don't know what's going to work, right? So they tried this dirty mix of contaminated vitamin D, and it turns out that the contaminant delivered beautifully inside the brain. These molecules stay in the brain for days after initial delivery, probably just because they're larger than the smaller siRNA. The monomeric siRNA gets excreted very quickly. Anyway, really exciting. And again, one dose gives 6 months to a year of knockdown in the brain. So this is very tolerable for patients, even though it's a rather invasive type of delivery. Oh, and I forgot to mention this company, Comanche. This is another, example of a bedside to bench discovery. And this is literally bedside, because one of the co-founders of this company was lying in her hospital bed. Her name is Melissa Moore. She's now the CSO of Moderna, who recruited her away from us, but she was a member of the RTI. She was pregnant, and she got preeclampsia. This is a condition of pregnancy where there is no treatment other than delivering the baby. If you don't deliver the placenta, the mother could die. And so it's a very, very serious condition. It causes a great deal of maternal mortality. It's the major cause of premature babies. And so Melissa is lying there in her bed, trying to get her baby to live, long enough inside her so that she could be viable. And the doctor comes in, and he's like, starts to tell her, you have preeclampsia, and she's like, yeah, but what's the mechanism? What is it? What causes this? And he was the co-discoverer of the causative gene that drives preeclampsia. It's called a soluble form of the VEGF receptor. SFLT1 is the name for it. And she's like, no, you tell me exactly what's this gene. And he described it to her. How is it made? Well, it's the VEGF receptor is a master regulator of endothelial growth. And of course, as you know, the placenta is full of blood vessels that supply the baby. It's an organ made by the baby that interfaces with the mother's vasculature. And in preeclampsia, the placenta starts to secrete a soluble form of the VEGF receptor. And Melissa is a splicing pro. It's alternative splicing. The reason the alternative message, the alternatively spliced message produces a truncated soluble form. This is a receptor that normally sits in the membrane, but the soluble form is released and goes into the mother's blood supply, and that's what's wreaking the havoc in the mother's system. So the mother's going to die because the baby is making too much of this soluble VEGF. And so, but the beauty of that target was that the alternative 3' end, the alternative splicing that creates this soluble form, has new territory inside the mRNA. The soluble form has an mRNA with a UTR, an untranslated region at the 3' end that is not shared by the receptor. And so where all the monoclonals, et cetera, that target the VEGF have failed in clinical development, the siRNA could go after the 3' end of the soluble form of this molecule without touching the receptor itself. So the beauty of it was that the drug could go directly after the disease-causing allele without touching or harming the natural and very important receptor itself. And so we started this project that led to this company called Comanche. Comanche has just one drug, and that drug is going to target preeclampsia. And the funny thing was, we tried to form Comanche before we tried to form Atalanta. So Atalanta was something we were thinking of forming, but this one we really wanted to get out there because it's a simple thing. It's one molecule. We think it's going to work. The baboon data was beautiful. All this data was published in Nature Biotechnology, and nobody wanted to touch it. Not a single investor would touch it because it's pregnant women. And who wants to be injecting anything into a pregnant woman, right? Nobody. But of course, the women who have preeclampsia want their babies to live. And so, there was a real need for this, but we couldn't get any investors. But luckily for us, right about the same time that we were failing to get investors for Comanche, Novartis bought the medicines company for $9 billion. That's the company that makes the inclisiran drug. And just based on that one drug, Novartis bought the whole company for $9 billion. And what was really fortunate for us was that the clinical development team at the medicines company was out of a job. And they came to UMass and they said, "What's new?" And we said, "Well, we've got this great preeclampsia target." And they joined our team. So they formed Comanche. And they are amazing because they just did this, right? They just did the biggest clinical trial for an siRNA therapy in history. They sold their company for $9 billion, and they came to us and they said, "We want to do preeclampsia." And the investor said, "Okay, here, let's write you a check." That was great. With investors loved them, right? Because anybody who just sold a company for $9 billion, you can raise $36 million easy. So they got the preeclampsia drug. It's going into women this year, and very likely they already have a buyer for Comanche. If that drug works, we'll know about 3 days after the first injection because the blood pressure will lower, and the mother will be able to carry that baby to term with just one injection of this drug. It's like I just can't wait. It's so exciting. And anyway, here's another one. This is another UMass company. Again, another colleague, a physician scientist at UMass. This is how all these companies have gotten started. Comes to Anastasia and says, "My patients have this autoimmune disorder called vitiligo, can you deliver to the skin? And Anastasia is like, well, probably the skin should be easy, right? And so they started a project and they formed another company, Aldina Therapeutics, that is delivering siRNAs to the skin to treat skin disorders. And, there's a lot of opportunities there. And an organ that, is easy, very easy to access. So that's another company. And then Diana Therapeutics, this is a new one that we hope will deliver to the muscle, the lung. I'll tell you a little bit more about that. Kidney, the ocular delivery is very easy too. So then types of tissues, the numbers of tissues that you can deliver to continues to expand. And the reason it's expanding is because, the team, not only at UMass, but elsewhere, they're screening for new conjugates and new modifications that enable these molecules to go to new tissues and to be more stable. And I'll just give you, when I get going, you guys might be here for another 2 hours at this rate. Thank you. So the chemistry space is huge, right? And it's very empirical. We don't know what's going to work. I'll just give you an example. So traditional molecule with a phosphorothioate, this is a type of linkage modification, is stabilized against enzymes that are degrading from the 3' end. This is an idea that Ken Yamada, a chemist in Anastasia's lab, but we recruited him to run his own group now. He's a brilliant chemist. He somehow decided that putting an extra carbon atom in the phosphodiester linkage might be tolerated. And so he did it. And this thing is like a rock. It's so stable that, and it works. It gets loaded onto RISC. It's tolerated. In the context of a phosphorothioate, it really is super protective. But you can also reduce the number of phosphorothioates, and phosphorothioates cause some toxicity. So lowering the amount of phosphorothioate is a good thing. And so far, this, what we call an extended nucleic acid, is well tolerated. It doesn't cause any toxicity that we can detect, allows you to have a lower dose. And you can see these are different parts of the brain. And we're looking at mRNA levels here. mRNA knockdown is beautiful. And this is after, I think, 3 months of delivery. So you can see beautiful knockdown in all these major parts of the brain. The deepest part of the brain is the hardest, but you can get a knockdown in this region as well. And at the protein level, it's even better. At a protein level, if you do a Western blot, the Huntington gene is undetectable in mouse and monkey 3 months after the first dose. You cannot detect the protein at all on a Western blot. The mRNA is always detectable because there's some nuclear mRNA that's always present. So you always see some mRNA, but if you look at the protein, nothing. And it's durable knockdown for months. So this is the kind of data that led to the formation of Atalanta. But these molecules, this has never been in a clinical trial. It's never been in a human, but we expect, based on the animal data, that it will dramatically extend the knockdown and the potency. One of the Achilles heels of an siRNA is that it does not have a cap. You know, the mRNAs are capped to protect their 5' ends. siRNAs have a 5' phosphate that's important for the molecule to be functional. That 5' phosphate is easily removed by enzymes in the cell, and all of the approved drugs that are in humans have no protection at all for their 5' end. This protection, called a vinyl phosphonate, which is basically a double bond that's put into this linkage here with the first phosphate, this is quite protective. It helps a lot. Again, hasn't been in humans, but we already know that when you put a vinyl phosphonate on an siRNA, it dramatically enhances the potency. So we do think that siRNAs, even targeting the liver, you can probably reduce the dosing to once a year once you've incorporated this 5' end protection. But that's not the end of the story, because again, Ken Yamada, and I can't show these structures because the patents are pending, but he's been playing with the 5' end and wasn't happy with the vinyl phosphonate because it wasn't stable enough for him. And so he has these new modifications that are like, again, they're very, very stable, and they still get loaded onto the Argonaut. They still seem to be fully functional, and hopefully we'll be able to talk more about what those actually are in the near future. A lot of this work, I mean, the chemistry group at UMass has completely eaten our entire budget for lawyers because they are filing like crazy. This is incredibly valuable technology. And, you know, again, it's something probably you have great chemists down here in Brazil should team up with them and start making new molecules that can be plugged in here, because there's a lot of space to study. So when the pandemic broke out, you know, here's a picture of the RNA Therapeutics Center. Everybody had to leave the campus, right? Nobody's allowed to work even. In the US, we were completely locked out of the labs unless you were working on COVID, which I thought was kind of silly because it's like that's not the only reason people are dying, right? I mean, there's a lot of other diseases. But anyway, so the siRNA, RNA interference groups, chemistry groups, decided to try to deliver to the lung and see if they could get a ionophore that would go there. And, you know, it turned out that these divalent siRNAs were taken up quite well in the lung. In fact, so were trivalent and tetravalent. One of the things that's interesting about these two is from the standpoint of the FDA, if it's a single molecule, it's a single drug. So as long as you can have a rationale for why, you can do one clinical trial, but you could potentially have 3 different siRNAs connected here, or 2, and that could be considered a single drug for the purposes of a clinical trial. But for some reason, these actually deliver quite well into the lung. This just shows an example of how, compared to a monovalent, you have very nice distribution in the lung. I won't go into the details, but delivers to quite a few different cell types in the lung. The knockdown is actually quite good. And so so when you want to make one of these siRNA drugs, what you do is you make a lot of them that target each gene. And here you're looking at the genome of the coronavirus, and you basically just screen each gene. You make lots, like 100 different siRNAs for each part of the virus, and you look for siRNAs that knock down efficiently. And of course, then you want to choose the ones that are in the least variant regions of the virus, the regions the virus can't change or probably isn't able to change because there are conserved functions encoded there. And so they found a bunch of leads. You can see some of them knocked down very well. And these are what you want. You want really high levels of knockdown. And then once those were identified, they did a cellular assay, and they could show that they could block the infection using the siRNAs. I won't go into the detail. Again, then they moved into animals and showed that you could get knockdown in the lung in an animal. And ultimately, they did a challenge where again, you can see this is just highlighting some of the different modifications. These molecules haven't incorporated the XNA yet. They're using phosphorothioate. Again, the chemistry keeps evolving. So, but this paper is just coming out now in PNAS. They get protection in this system from infection. And the way this would be used is not to treat somebody who's infected, because there's a delay, too long of a delay. Once you give somebody this, they will hopefully be resistant to becoming infected. And then their lungs, even if they get infected nasally or in the rest of their body, their lungs would be protected against the virus replicating in the lungs. As you can see here in the mice, the plaques don't form when the drug is being administered to the mice by inhalation. And so you could imagine a setting where healthcare workers, or let's say sailors on a ship who have an outbreak, get a dose of this to try to prevent them from getting a very bad sort of infection in their lungs. And we got a lot of funding to do this project from the US government because, you know, obviously you can make these molecules very rapidly, even faster than making an mRNA vaccine. And they have the potential to, you know, be, you know, long lasting as well. For months after injection, a person might be resistant to developing a very bad lung infection. So another thing, and this is, you know, just an aspiration, cancer. You know, we can deliver now beautifully to so many tissues. And I didn't talk about it, but beautiful data on muscle delivery, heart delivery. You know, there's really, I think, exciting opportunities to make these molecules for any normal tissue. But what about cancer? Nobody's even tried. I mean, there've been a few tries to get cancer siRNAs working, but not with these new chemistries. A lot of the failed attempts were with insufficient quality molecules. So going after cancer, this is something that we want to do systematically at the RTI. We want to like start making, bringing in models, but also giving out siRNAs conjugated in different ways with different ionophores attached to them, and start exploring delivery into cancer, because cancer is a great target. Cancers have their own genetics, you know, because of all their mutations. Sometimes they're trapped in certain genetic pathways that they really need to use, and we can go after those. So again, the future is really bright for this kind of molecule. I'm going to stop. This is my last slide, and just tell you that this mechanism is really, really ancient. And what my lab is really fascinated by right now, I don't work on therapeutics. My lab is more interested in sort of the basic biology, the fundamental biology. This is showing you the ovary of the worm, and these are the eggs that are forming, and these are the nuclei. That's where the DNA is. The green stuff is mitochondria, which is so cool. Look at it, it's flowing. So there's a factory of in the ovary, the immature gametes that are in Pachytene up here are like a factory. They're making gene products that get loaded into the eggs. The eggs fill up. And one of the things that you can see are these red things. These are what we call nuage. And the nuage form this beautiful perinuclear cloud around each of the germline nuclei. And you know what's in there? More than 1 million different Argonaute guide complexes. The human germline also has millions of these. And I told you they're involved in transposon surveillance. This is true in humans, plants, fungi, all animals, all, you know, worms and humans, in fact, use the same sort of group of Argonauts, this peewee group of Argonauts that control transposons, but they're doing a lot more than that. In fact, they not only control the transposons, but they also control the chromatin. They're directly, these are nuclear silencing pathways. The RNA goes into the nucleus and controls the chromatin state, whether the gene is even turned on or not to express an mRNA. And all of this is going on in the germline. And then these millions of guide RNAs are loaded into the nuage, and those get incorporated into the egg. And what these guide RNAs, we think, what they do is they reprogram the chromatin again in the zygote in the next generation to essentially reestablish, to reconfigure, to update the software every generation, you know, and the animal is actually sending information from the parent to the offspring, telling the offspring which genes should be turned off. And you know, this phenomenon of transgenerational inheritance is one of the things that really fascinated me from the very beginning, because when we started injecting RNA in the animal way back in the 90s, early 90s, what really surprised me was that we could induce silencing in the mother, and the progeny would have silencing, and then their progeny would have silencing. We could get these long-lasting, durable, transgenerational effects. And in fact, I think this is really what it's telling us, is that RNA is still a master regulator of inheritance in all organisms. RNA, I showed you the RNA world where RNA was in control, and they created DNA to be like its slave, right? The DNA is much less important than the RNA, right? It's just the storage, it's the hard drive, right? So the RNA, I think, is what this is telling us. The RNA is really controlling a lot more than we thought, the information that's fundamental to life. And so we're trying to learn the rules for how that works, trying to learn more about it. I think there's a potential someday for employing Argonauts to mediate chromatin silencing on targets. And so that's a really exciting thing to be thinking about for improving maybe a new future kind of drug. And I'll stop there. Most of the work I talked about was from Anastasia and John's chemistry groups, and Julia Alterman actually developed the dimeric siRNA story and is now an assistant professor at UMass. We've recruited her. She was a grad student at UMass, and she kind of skipped a postdoc and went right to having her own lab. She's got millions of dollars of funding already. And Ken Yamada is the chemist who developed the XNA and also the 5' phosphate stabilizations. And all of them are recruiting. They would love to have people from wherever who have an interest in making this kind of molecule, especially chemists who are hard to find. There's probably a lot of chemists here, and maybe not in the room, but we're looking for you guys. You're really important. It's funny, after the Nobel Prize, right after that big dip when everything was crashing and nothing was working, a reporter called me up and she said, "It's been 10 years and there are no drugs. What would you have done differently?" And it was one of those gotcha questions, right? Because you have to admit it's not working. And I told her, and I wasn't joking, I would have hired more chemists, because that's what we did. We hired these chemists, and they've really made a huge difference. And I think the future is very bright for this kind of medicine. And you should all start thinking about it, both making it at home, because eventually you will be able to, everybody will be able to, and everyone will need to, but also to get involved in discovering the next thing. Who knows what else is out there to be discovered in how organisms handle information. It's really a huge amount still to learn. And I'll stop there. Happy to take any questions.
All right, we can have the lights. Thank you. So we have plenty of time for questions, and I want to also tell people across the room with the 20 seconds delay that they will hear me from now, but you can come across here and make questions too if you want, okay? Those in the Anfiteatro Azul can come here as well. So questions, please. Oh, I must say, if you want to pose questions in Portuguese, we can translate, okay? So,
what is the major challenge in the RNAi field to find the best target genes to make functional RNA drugs themselves, as you mentioned, because of the chemists, or to perform the on-target RNA delivery? Because thinking about cancer and this kind of other.
Yeah, what I think one of the biggest challenges is that it costs a lot and it requires a lot of expertise to develop a molecule that targets a gene that you might be interested in, right? So you, as many of you know, you can buy siRNAs pretty cheaply, but those won't work in an animal, right? So there's no way you can use those to study a gene. So the bottleneck, I think, is that the therapeutically relevant molecule is difficult to make and it's costly to make. And when you make one, it's so valuable. So you can have a company like Alnylam or the medicines company, they make one molecule that works on lowering LDL and they can get $9 billion, right? And so they don't really care about making 1,000 or 5,000 of these molecules because they can make one and they'll never have to work again a day in their life, you know? I really do think that's part of the problem. So we're trying to make this company, Diana, that will make hundreds of these molecules. We want to make the Amazon of siRNA that will make these molecules and give them away to people like people in this room, because everybody has targets. And the part that you can scale is making the molecules. So we can make these, we can patent them, and then we can give them to you. You can show if they work in your disease model, if they target cancer, if they target whatever. You can do that at essentially no cost for the reagent. You just have to have a credible idea that can be tested. We could make hundreds of these every year. Once you've automated it and scaled it, we can make lots of these. We can make one for every gene in the whole genome, the whole human genome. And in the future of these molecules, we'll be using them in combination, not only with each other, but with other existing therapies. So you'll be using for cancer therapy, of course, you'll still be using, you know, whatever immunotherapy, whatever is the hottest thing, but then you'll be also using an siRNA because you don't want the cancer to come back. So you're going to be hitting it a little extra hard. This is that's sort of my dream, is to try to democratize access to these molecules, because the huge bottleneck is the cost of running the animal studies. Making the molecules, even though it's too costly for an individual laboratory because you don't have the machines, you don't have the chemistry expertise, if you scale that part up and generate, you know, lots of them, and then distribute them to people who have the animal models, that's how I think we're going to break this open to make lots of drugs and to really speed the discovery process. And then, you know, it costs a huge amount of money to bring one of these molecules into the clinic. So for preeclampsia, they raised $36 million. It's a relatively straightforward clinical trial. They're going to have to raise more before the clinical trial is done. But you know, that's just for one drug. You know, 36 million gets you into the clinic, but doesn't get you all the way through the clinical development. And so, yeah, that's a great question. I don't know if I really answered it, but.
Hi, Craig. First of all, on behalf of our institute, I'd like to thank you for sharing all this brilliant talk and beautiful data. And my question would be more on that cellular point. When you showed us the data on the liver and that you have that stabilization, do you detect any kind of transfer from cell to cell in extracellular vesicles? Because you said that it was stable in the lysosomes and it was quite a long time to have the effect.
Yeah, that's a great question. The literature on extracellular transport of microRNAs and siRNAs is really exciting. I mean, clearly it's true in C. elegans, and there's a great deal of data actually in vertebrates as well, and even in humans. There's data suggesting that microRNAs are transported in little vesicles that are in circulation, and the liver produces these for sure. You know, the immunological synapse is a place where a lot of these RNA molecules might be getting transferred. You know, even mRNA is thought to sometimes be transferred by direct, you know, cell, almost, you know, vesicle mediated transfer. But so the question is, you know, to what extent does that play a role in the therapeutic setting? Not really clear. Whether you get broad systemic distribution from liver delivery, that doesn't seem to be, at least at a therapeutic level, but I know that there's a great deal of interest in sort of natural microRNA secretion by the liver. There's some really exciting data that miR22 is a master regulator of metabolism, is secreted in vesicles from the liver, and that might be having an effect on peripheral tissues like fat. So yeah, it's really, I mean, I think if anything you take away from this talk, just, you know, it's humbling how sophisticated organisms are and how little we actually know about what they're doing. Like they're incredible, and they are really, they have a lot of tricks and mechanisms that we have not even begun to understand. So yeah, you have to approach, I think the future of biology is going to be punctuated by many new discoveries. And I think the systemic transfer of information within the somatic tissues of an organism is going to be fascinating. It's clearly played a major role in the evolution of life, and probably transfer of information between cells, including both mRNA and microRNA, and maybe some other kind of RNA, is going to be important too.
Thank you, amazing talk. So innate immune response has many receptors for nucleic acid sensing. So I'd like to hear from you, when you develop those molecules, are you also thinking about on ionophores, either to escape this sensing or either to boost it, depending on the scenario?
Yeah, that's a great question. In fact, the initial failure of a lot of the drugs that were tried without any modification was in part because they were stimulating that innate response, you know, cytokine response, for example. And so that's something to be avoided at all costs, probably in most settings. You know, maybe in a cancer therapy setting, you know, it would be a boost. But in the case of these drugs that are now working, it turned out that placing modifications into the backbone and the base, you know, the 2'-O-methyls and 2'-fluoros, and those actually dramatically reduced innate immune stimulation, and that was actually one of the most exciting reasons for putting them in, not only for the stability, but also because they seem to block that. And I'm not quite sure what the molecular or, you know, the structural basis for that is, but that's the case. The practical finding is that these chemistries can also avoid stimulating the immune response. Thank you.
Thank you very much. Very nice talk. We are going through a political and scientific crisis in Brazil, so it's really great to have you here and motivate us. My question is about mitochondria. So any of these drugs, I'm sorry if I missed it, do they target mitochondria genome silencing or mitochondrial diseases?
Yeah, so first of all, we're having a political crisis too. I don't know if you guys have noticed. I think it's spreading over the whole world. But thank goodness scientists are such sort of good people generally. You know, I think probably most of us think of ourselves not as Brazilian or American, but as human beings. You know, what a concept. You know, we're actually all in this tiny little planet together and share so much history. Mitochondria are you know, really fascinating. A lot of mitochondrial gene expression is now derived from the nuclear genome, and of course all of that is readily accessible to an siRNA approach. Getting siRNAs to go into the mitochondrion hasn't been solved to my knowledge. It might be possible. I'll never say it's not, but I haven't heard of anybody doing it routinely. So that's going to be a challenge.
Hi, thank you for your talk. I'm going to ask 3 questions. Like about the costs you were saying, is it even feasible to pursue that kind of drugs for neglected diseases? And are there studies targeting RNAs from pathogens, not viruses, that you showed? Or is it better to target host responses to like host-directed therapies? And can you comment on organisms that does not have conserved RNAi, like some leishmania species? Like,
Wow, there's a
Thank
lot
you.
of questions there.
Sorry.
You might have to keep the microphone because you're going to have to remind me. So in terms of targeting parasites, for example, again, it's a challenge, that hasn't been addressed. Can you deliver to a microbial cell? First of all, most microbes don't have argonauts, so can you modulate an infection by modulating host genes, the vertebrate human genes that the microbe needs? Surely, yeah. There's a lot of ways that you can. I think almost any type of disease, whether it be driven by a virus or a parasite, could be addressed. Certainly, the easiest thing would be to address it by looking at the host gene expression, because once you start trying to deliver into another organism, let's say it's a nematode infecting a gut of a human, how do you get the siRNA into the nematode? If that's what you want to do, yeah, maybe you can do it. So there's a lot of challenges, but I wouldn't say it can't be solved. And the other questions, was there a question about the cost?
yeah, like for neglected diseases, everything
yeah.
like it's more difficult, like the funding and like
Yeah,
after like
that's one of the
drugs.
reasons for this company, Diana, right? Because the orphan diseases get ignored. Because if you can make $9 billion by lowering cholesterol, why are you going to study something or help make a drug for something that has only maybe a few people a year get it? We have a therapy for a form of ALS that has only a few people infected per year. As you know, many different diseases now, we know, like ALS or cancer, there's so many different types and they're genetically distinct. So when you make a genetic drug, it won't necessarily work for all of them. It may only work for one. And then that cost of doing the clinical trial is still very high, right? And so companies are not going to make those, as their first choice. And so one of the ideas for a company like Diana is that it will make the molecules, and it will, because it can do it fairly cheaply, and then it will let an investigator like yourself show that it works in a model for a rare disease, and then you can raise money from investors once you've validated the approach. Investors will still help you raise money, for one or two. That's the kind of the idea for trying to tackle that problem. It's a huge problem because most genetic diseases are kind of limited by the number of people who have that mutation. Another example is Alzheimer's disease. And one of the things that's so exciting also about diseases like that, and including cancer, is that we are learning a lot from healthy people who don't get dementia. For example, we have genome data for them. And so some of the genes, it might be possible to go after orphan diseases by modulating genes that improve the ability of the cell to deal with cancer or Alzheimer's. Neurodegenerative disorders could be ameliorated by, for example, improving autophagy. Upregulating autophagy might help a lot of these diseases that are caused by aggregates, et cetera. If those aggregates could be digested away by elevating autophagy, you might not need to block the Alzheimer's gene, and there would be a lot of them that you'd have to go after each one individually, but maybe you can just upregulate the other pathway, that sort of master regulator. So there's a lot of ways to try to solve those kinds of problems.
Thank you.
You're welcome.
Thank you for your presentation and your presence here. I would like to escape a little of the topic, but I think the main question around us is how did you feel when you back home after won a Nobel Prize?
Well, I'm a very humble person, and I know that I was very, very lucky because I was working on this beautiful little animal. And the only thing I did that was really smart was I chose not to work on humans instead of, because it's so hard to do these kinds of experiments in a human. I never, ever, ever could have discovered something like this by working directly on a human. But because I knew the genetic code was the same in all animals, including bacteria, I didn't care. I figured it's an animal and it has a short life cycle and probably easier to study. So that's why I started trying to inject DNA into that animal. I was like, I'm going to put DNA back into an animal. It had never been done. And it was a lot easier to try it on a worm than on a human. So I lucked out, I just chose the right animal. And, and then when you get the prize, of course, it feels like the universe is fake. How could this happen? And they throw an amazing party in Stockholm, I have to tell you. Definitely. If any of you win, invite me to your party. I want to go.
Thank you for nice talk. You mentioned vesicles to deliver different RNA, and I was wondering if you are interested in look directly to extracellular vesicles and deliver this interference RNA to treat different diseases.
Yeah, a lot of people are working on trying to understand the natural extracellular transmission of these signals inside the human body. That's not what I work on. From a therapeutic perspective, if you encapsulated them in some sort of a natural vesicle before administering them, you might be able to enhance the systemic delivery. So that's a very exciting area. There aren't any breakthroughs that I'm aware of yet using that approach, but maybe someday there'll be some way to formulate siRNAs for even better systemic delivery by encapsulating them in some natural, vesicle type thing. Yeah, a lot of people are working on that.
Okay, sir. Thank you for this great lecture. So my question is, for the delivery of this divalent siRNA, have you ever tried using nanoparticle for the delivery? Then the second question is the RNA interference. Is it a form of epigenetic regulation?
Yeah, so the answer to the first question is that the concept for most of the technology that's both succeeding in the clinic and that I'm aware of in development has moved away from encapsulation. For mRNA, you definitely need to encapsulate them because it's a big molecule and it's mostly unmodified. So the mRNAs would be very unstable the way they're being manufactured now. The vaccines have to be encapsulated. But for these siRNAs, they're now so stable from the linkage modifications and from the base modifications that you don't need to encapsulate them. You can just put a ligand on them, and then when they go to the cell, for example, with GalNAc, they get endocytosed by the cell and get taken up. And that's also how the CSF is probably taking up these dimeric molecules. They're not encapsulated to anything. They're circulating in the cerebral spinal fluid, and the cells throughout the brain are engulfing the CSF constantly. That's partly how they get their nutrition, and they're taking these molecules up constantly. So we don't need to encapsulate them, which is great because encapsulation is, it's a whole science. There's nanoparticles, very sophisticated science there. A lot of immunostimulation from particles can be a problem. So it's nice that we can not have to do that. And then the other question was the second question? Epigenetics. Oh, yes. Yeah. So that was one of the things I mentioned. We're we noticed that they could transmit silencing to offspring. We now know that the Peewee system, the pRNA pathway, is involved in that transgenerational inheritance. This mechanism that humans also have. It's another Argonaut system that's expressed in the germline, and this system is involved in programming heterochromatin that maintains the silencing of the transposons. So yes, it's a major mechanism of epigenetic inheritance, and this is true in every type of organism. So plants, fungi, worms, humans. So it's an important mechanism that we still don't know that much about. There's a lot more to learn about how that all works.
So thank you for the talk. It's really nice. I have two questions. The first is whether you're looking for a way to deliver these RNAs into enterocytes, maybe through oral delivery. And the other is more regarding how to make finding the right sequence more easily. So maybe you maybe have a modification, a chemical modification, so you can have more on-target effects and less off-target effects.
Yeah, so oral delivery would be fabulous. And then one thing that looks like it actually has worked is you do need some sort of encapsulation for oral delivery. And one of my colleagues has been playing around with yeast particles, which are basically the remnants of yeast. They're the shells that when you make beer, you can get a lot of this apparently. I think Brazil is one of the biggest exporters of yeast particles. They're like the leftovers from lots of agricultural applications. But anyway, you can stick stuff inside the yeast exoskeleton, the shell, and it can be encapsulated there, and then it can get through the digestive system. It can get taken up. And so there was even a Nature paper saying that worked for siRNAs many years ago. It doesn't mean it's not true, as most things published in Nature turn out to not be true later. But it still needs a lot of work. It'd be so great if you could just orally take it. But then the nice thing is the sub-Q injection is a great delivery mode. And I have a diabetic daughter, so she does sub-Q injection every single day, right? It's not that hard. And if you only have to do it once or twice a year, it's super easy. And then the other question on target. Yeah, so a huge amount of computational stuff goes into the upfront part, because when you're, let's say, designing siRNAs against COVID, you have the sequence, right? But right away, that some of the sequences would not be a good target because they're too similar to a human gene, right? So you want to avoid matching the human gene within this part, the first part of the guide, which is called the seed, because the way the guides work is they use the first part of the guide first. And if it matches, then they do more. But even just matching 7 nucleotides could give you some silencing, right? So this off-target issue is a big issue. And so that can be solved in part by computational approach. And then once you've done that, then you don't even bother synthesizing those. And you still have hundreds of opportunities because you have all the rest of the sequence, So usually you don't have to, you can computationally eliminate a lot of that. And then you can also do, the field has just matured so much. The passenger, they call it the passenger strand. There's the guide strand. And then because it's a double-stranded RNA, the configuration of the drug initially has 2 strands, but you can completely prevent the other strand, the passenger strand, from ever entering the Argonaut. So there's a lot of little technical tricks for eliminating what used to be a big problem for us with off-target. So they're all, I think, pretty well in hand, and the computational aspect is good, but it's not enough. So you still need to make lots of them for any given target. You can't know for sure that this is going to be a good guide RNA until you make it. And I think it's because of the complexity. The mRNA molecules are base pairing with themselves and with other molecules, and there are proteins binding to them. They're not naked RNA, and you don't know where those interactions are normally in your cell. So if you know, what might work beautifully as a guide RNA on one RNA wouldn't work on another because there's a protein that's sitting right next to that spot, or it's folded up and not accessible. So it's more empirical at that point.
Greg, we have like lots of raised hands, but we are running fast time. So what I propose, I will let one last question in order, and that will make us, you know, topics for the session in the afternoon. So I remember students, I will address them in Portuguese, just a little bit. So, pessoal, temos a sessão às 2 da tarde. Vocês podem perguntar para ele o que vocês quiserem, tanto com relação à apresentação, quanto sobre curiosidades como do Nobel, ou do que vocês quiserem, tá bom? Então, vou deixar só mais uma última pergunta, porque senão a gente vai estender demais. Tudo bem? So, last question.
Hi, Professor. I'm so nervous. It's a pleasure to talk with you. And I have a biotech company that develops microRNA for treatment of chronic disease. And always we talk about RNA therapy. The FDA, big pharma investors ask about the security profile. So I'd like to know, in your opinion, what's the best strategy to identify and protect that of target's effects?
So you're saying that investors are worried about off-target?
Yes, yes.
You're having a hard time with that? And is your company using a natural microRNA?
No, chemistry modification in microRNAs.
I mean, one of the concerns that you might have, a lot of companies have, there have been whole companies that failed, you know, trying to become a microRNA drug company. And one of the problems is microRNAs have evolved to target thousands of genes, right? I mean, that's why that we have them. So if you use a natural microRNA as a drug, you might have to worry that it will be targeting things naturally, many things that you don't even know what they are, you don't understand. I mean, the understanding has grown a lot. So I'm not sure exactly why the investors are concerned. There could be valid concerns. If you're designing an siRNA that's not a microRNA, that it's not meant to mimic a microRNA, but rather to silence a gene, it gets loaded onto the same Argonaut that does the microRNA silencing, but now it's a totally new sequence. It's not a sequence that evolved naturally to target genes. So you've made it, and so you get to pick it, and you can choose which ones give you the least off target. So that's actually just normal part of the pipeline now. You know, of course, you'll knock down gene expression, you'll do some RNA-seq, mRNA-seq, you'll look at which mRNAs got knocked down. Hopefully your target got knocked down and not, you know, 100 or 1,000 other genes. And then you use that data to convince yourself that you've found a good molecule, and then you can move on to the next step. So that's what we do.
Okay, thank you.
You're welcome. All right, well, thank everybody. I look forward to more discussions, and I'm spending the whole day with you guys.
Yeah, so we have plenty of room in the afternoon.
Yeah.
All right, I want to say thank you once again, Craig, for coming.